And we've already pointed out that we have a tool which is available in this course called PlotViz, and I've shown you PlotViz and applied it to the results of clustering of when we were discussing recommender engines and other types of um, other types of applications. And so we have a variety of different tools here. Multidimensional scaling is the simplest example. We have our own improvement of that called deterministic annealing multidimensional scaling. We have these work for general problems. For the cases where the data, which is not true for genomics, where the data comes from a vector space, we have algorithms like generative topographic mapping and their improvement with deterministic annealing. So there's a rich amount of work on information visualization, which I expect to get more important as we get more and more high dimensional data. And we're sort of desperate to analyze it. And I think this basic idea of using Google Earth or Google Map type interface, where you map everything to 2 or 3 dimensions and then browse it, is likely to be important, because when you do this mapping, it's done so distances are preserved, so sequences which are near each other in the mapping will be near each other in the real space, and so they're likely to have some similarity. So this browsing is a way of collecting related sequences together and looking at them. And this slide here points out actually how you do this particular multidimensional scaling. For those familiar with the chi-squared concept, it's basically chi-squared. You map your sequences to points xi, and xi, sequence I goes to a point xi in 3 dimensions. You calculate the distance between sequence I and sequence j in the original space. You apply actually a monotonic function to that calculation to make it have better properties. Then you take this this modified distance, you subtract it between the distance in the map space, take the least square sum, you normalize it by 1 over the distance, so you enhance the importance of small distances, which are usually the most important, and you just minimize this the function of xi. So we did this in one case up to a couple 100,000 points was the largest one we probably ran up to now. So that's 600,000 parameters sitting here to be optimized. Here's some examples of this is not proteomics. This is not the protein universe. This is the genomic universe or metagenomic universe, and these are so-called 16S rRNA samples, and these are just 100,000 sequences clustered. That's the coloring, and mapped to 3 dimensions. This clustering is not complete. It needs further clustering. It's just clustered to a certain resolution so that you saw some structure in the problem. Here is the actual 680,000, which is gotten by taking that original set and interpolating the remaining data in the set to that. This interpolation, we now know how to do better on some of these glitches like this. These little circles here are due to mistakes in the interpolation. They don't affect the broad picture, but they affect the answer in detail. Always in this world, you keep improving things. So in this monotonic function, we try to choose that monotonic function to spread out that distance and make it have a broader range, because if you have a broader range, you'll get better mappings. If the distance is such they're all very large and don't vary very much and they're all roughly the same number, which is what happens with the raw data, you'll get rather pathological mappings which don't look as good. And as long as we choose a mapping function f, which is monotonic, we do not change any of the, we preserve what we want to do, because it is not, there's no obvious reason why the raw distance is the one we map to the distance in 3 dimensions. Any monotonic function of that raw distance can be used. I pointed out that with this MDS is order n squared, but we get around that by producing very fast interpolation routines, which we're getting better and better at. And then we have various ways of doing the MDS. either by the so-called Smakov method, which we're gradually improving, or by just standard chi-square minimization, where we use the so-called Levenberg-McQuark method. And then we go and visualize all of this in this program plotviz that is attached to this course. So the original data for this proteomics case was 100,000 sequences. We later increased to 180,000. We calculated that there are various ways of calculating distances. One is using Needleman and Wunsch, and that is the one we found most reliable for this problem. Sometimes we use other methods like Smith-Waterman Goto distances, which for this, whereas Smith-Waterman I think is better for metagenomics. For these proteomics, the Needleman Wunsch gives you better answers. Then we do a form of function f, which I don't want to go into, which actually we use the so-called square root of 4D method. Go to our paper to see what that is, and that defines this mapping function f. And if you look at the formal dimension of the distance data, for the raw data it's 244, and then the transformed data is 14. Then we take that data, which is of dimension 14, and then we map it to actual three-dimensional vectors. So we keep going down, but 244 to 3 is a bigger step than 14 to 3. And you'll find that the resultant mapping more uniformly covers the Euclidean space if you do this. Here's this proteomics data, and it is quite amusing handbag shape. And we've shown a few clusters which show that these are clusters coming from the experimentalists. Obviously, a lot of these are truly clustered. In fact, they're all clustered. Some of these clusters, like this white one here, are really not so solid. I mean, they're not really super well clustered. Others are like this, and some of these ones here are pretty close to each other. So here is a little comment on the general technology of minimizing sums of squares, which is called the chi-square problem or the least squares problem, where chi-squared is the sum over a set of measurements of the value of the measurement minus the prediction of the measurement all squared over the error in the measurements all squared. And I say for multidimensional scaling, the value of the number of points is n into n minus 1 over 2, when n is the number of points. So for n of 100,000, this is 0.5 times 10 to the 10. So this is a pretty big sum, and so that's why you desperately need parallel computing, because you can calculate this sum in parallel and get huge speed up. So all of these jobs are run on 1,000 cores, or so up to actually 768 cores, and we get good speed up there, and that's why it's feasible to do this problem. And chi-square, because it's a sum of positive numbers, actually has a minimum because it's bounded below by 0. You can never get it, so it's if you make the data value prediction very far apart, it's bounded above by infinity, you'll get huge chi-squared, and somewhere between somewhere there's an actual minimum, which you have to find. And as I said, it's pretty hard to minimize. You need some fancy methods to make that work, but it is naturally parallel over points and initial starting positions. Here's some comments about this problem. So we have a lot of terms in chi-squared, and the number of terms is much bigger than the number of unknowns. So there are lots of constraints to find the unknowns. However, if you look at chi-squared and just sort of think about it as a function of these 3n unknowns, there are going to be some combination of unknowns which chi-squared depends very slowly on. And those, it's very difficult to try to determine poorly determined parameters. So if I look at this problem here, it's these different shapes. This shape here has a pretty good minimum because it's got good curvature in all directions. This shape here, you can find where you are in this direction here, from between this curvature, but to find what's going on in this direction is essentially impossible because chi-squared hardly changes as you move this direction. And if you take a real problem, you have complicated shapes. Here we have a shape with lots of minima, and otherwise and to try to navigate these spaces and trundle down these slopes to find the right answer is pretty non-trivial. And that's what you need, all these fancy technologies where we use the Levenberg-McQuark method or the Smakov method to solve.